right, so today we're just going to go over the WISER review that you had on Monday. Sorry, we're going to go over the WISER review you had on Monday. Remember, there was also a Kahoot review I gave you yesterday. It's in Wednesday's folder. And then today you have another test review, and that's going to be your actual assignment for today. So again, we're going over the answers to Monday's review on WISER. So if you want to go ahead and open that up, we will look at that right now. All right, so for number one, you are going to match the terms with the descriptions. Before we start, I just want to talk about these phases. Interphase includes G1, S, and G2. Prophase, metaphase, anaphase, and telophase, those all belong to mitosis. And then cytokinesis and mitosis, they all belong to the M phase. And again, that one wasn't part of your review. I just wanted to remind you that before we start. All right, so the very first one is interphase. And interphase, it starts off with the parent cell. It's the DNA in the form of chromatin, and the cell grows and the DNA is copied. That all happens during interphase. In prophase, the nuclear membrane disappears. The spindle and centrioles start to appear, and the DNA is in the form of chromosomes. And those are going to be your X's. Don't forget, chromosomes are your X's. In metaphase, the chromatids are going to start to long up along the equator line. They're all going to be in the middle. In anaphase, the sister chromatids are going to separate. They're going to come apart. In telophase, the nuclear membrane is going to come back. Around the chromosomes, the cell plates are going to form in plant cell division, and the animal cells are going to start to form a cell furrow. And then last but not least, cytokinesis is the actual division of the cytoplasm. And that's where we finally have two separate daughter cells. Any questions on number one, matching up the phases? All right, let's keep going then. Number two, what is the end product of mitosis? Well, the whole point of doing mitosis is so that way we have two daughter cells. That's what we want. We start off with one, and it splits into two. And make sure that they're identical. We want the same number of chromosomes. We don't want half the number. That's meiosis. That's whenever we do reproduction. We're not there yet. So we want the same number of chromosomes. Any questions on number two? All All right, number 3, we're gonna match the terms with the description. Chromatin is gonna be our loosely packed DNA. That's where it kind of looks like spaghetti. Our chromosome is going to be our tightly packed strands of DNA. This is when they're organized into their X's. And a chromatid is a single strand of a duplicated chromosome. So this would be one chromatid. This would be another chromatid. Together they make a full X chromosome, but apart they're just single chromatids. Any questions on that one? All right, number 4, what is the correct order of the cell cycle? The correct order out of all of these, this one's going to be interphase. That's your G1, S, G2. Then you have mitosis and cytokinesis in the M phase. So your correct answer is B. Any questions on that one? All right, let's keep going. Number 5, what is the longest phase of the cycle? The longest phase is going to be interphase. That's where the cell spends about 90% of its time in interphase. That's where it's doing its normal functions. It's also growing, it's also getting ready to duplicate the DNA, and it's duplicating other organelles, all in interphase. Number 6, a great white shark has 82 chromosomes. When its cells undergo mitosis, how many daughter cells are made? It doesn't matter how many chromosomes it has. The question's asking about how many daughter cells. And we're always going to make 2 daughter cells during mitosis. Always. Any questions on 5 and 6? All right, let's keep going. So now we're going to match the following phases with their correct diagrams. So in prophase, that's where we have everything looking like X's. Our chromosomes are going to start to appear, and that's going to be this image right here. Remember, in this phase, the nuclear membrane is going to start to disappear, and the chromosomes become visible. In metaphase, everything's going to line up along the equator, along the middle metaphase. In anaphase, everything's going to come apart. So that's this image down here. Anaphase, apart. Then during telophase, we have 2 nuclei. The cell's not apart yet, just the nucleus. We have 2 nuclei that are separated from each other, but not the cell yet. And then finally, cytokinesis, that's when we have 2 cells. Any questions on the images? All right, let's keep going. We have 1 more page left. It was a pretty easy review. What structure holds the chromatids together? That's going to be the centromere. And that's that little tiny part right here. Centromere. What is the name of programmed cell death? That is going to be apoptosis. Again, that's when the cells start to shrink. The cytoplasm and the cell membrane start to form around individual organelles, and then another cell comes in and engulfs them. Number 10, the nitrogen bases of DNA always pair up the same way, match the correct bases together. Well, adenine is going to go with thymine, always together. That's how I remember that one. And then cytosine is going to go with guanine. And I always remember grandma's cookies. All right, number 11, in DNA replication, the term semi-conservative means it's going to produce 2 copies, and it's going to have 1 original strand and 1 new strand. So that's what semi-conservative means. If it was fully conservative, it would have 2 original strands, but it's semi-conservative because it has 1 original and 1 new. All right, number 12, in which of the following phases is the cell cycle and DNA replicated? So we want when DNA is copied, that's what replicated means, copied. And that's going to be the S phase. That's what we call DNA synthesis. Synthesis, replicated, copied, they all mean the same thing: to make something, to copy something, to replicate something. So that's all 3 of those essentially mean the same thing. Finally, on 13, the very last question of your WISER review, what is the gray blob representing? Well, the gray blob, if you can see it, it's coming down and it's tearing apart the DNA. And the only enzymes that break apart DNA are the enzymes known as helicase. So that would be C, and it breaks the hydrogen bonds. That's how it breaks apart the DNA structure, by breaking the hydrogen bonds. Any questions on this last page? All right, so just as a reminder, your test is tomorrow. I gave you this WISER review and your FRQ questions on Monday. So all of those are in Monday's folder. Yesterday, I gave you a Kahoot link. So that is in Wednesday's folder. If you want to go ahead and review that again, you can play up to 20 times, I believe. And then today, you have another review in your folder, and that's what needs to be completed today. So if you have any questions or if you need any help with anything, stay on. If not, you are free to go.